From his experiments on pea plants, Mendel discovered three laws to genetics. Since Mendel was so meticulous in the breeding and analysis of all of his many different pea plants, he was able to discover three common laws that govern the results that he found. The first law is the law of segregation. This means that two alleles of a gene that are found on different chromosomes segregate independently of one another into gametic cells. This is certainly an updated version from what Mendel actually discovered here. We are integrating it with some of the knowledge we have from molecular biology of how DNA and cells actually operate. So let's look at how that actually plays out with our understanding. Meiosis is a process where a single into different daughter cells, well, actually 8 daughter cells, and each cell has half of the complement of a full genome here because it's going to combine with another gametic cell. And when it's separating the two alleles at each different locus, it's random which one is going to get. Each individual cell is going to get. So it's interesting that Mendel actually predicted the function of meiosis before we even knew this was a thing. The second law is the law of independent assortment. This one says that genes that are found on different chromosomes are sorted into sex cells independently of each other. What he's saying here is that different traits or different genes are independent. So to look at this, now we want to look at two different traits and see how they assort with each other. So now we're looking at seeds, both their color and their shape. Are they round or constricted? If these were dependent on each other, we would expect them to sort the same way. So we would only expect to see round and yellow or green and wrinkled. But what we actually see is all combinations of the two. So here we have to do a more complicated Punnett square because we're looking at two genes at the same time. And that means there are 4 different possible combinations of these traits that the offspring can have. So just remember that when you want to look at more than one trait, your Punnett square might start to look a little crazy, and you'll get a slightly more complicated phenotypic ratio as a result. The last law is the law of dominance. And this is that a dominant allele will always mask a recessive allele. This one's probably the easiest to understand, and we already talked about it a little bit when we were looking at this diagram here. Remember phenotype as the physical traits, and genotype, that's the invisible genetic complement at a specific locus. So here we were seeing that the tall trait masks the recessive trait. And this was actually one of the very first clues Mendel got that we were looking at particulate rather than blending inheritance. So again, Mendel has 3 laws: the law of segregation, the law of independent assortment, and the law of dominance. The law of segregation means that alleles are independent of each other. Law of independent assortment, that's talking about chromosomes, how chromosomes are independent of one another. And lastly, the law of dominance state that some alleles will mask others. So again, here we've been talking about Mendelian genetics, and that just means any genetics that follow Mendel's laws. Of course, there's actually a lot of genetics that doesn't follow these laws at all. So some of the easiest to understand that do not follow Mendel's laws are some different systems of dominance. So here we have something called incomplete dominance. So a white chicken has a genotype of WW, a black chicken has a genotype of black or BB, and a speckled chicken, now this is our heterozygote. It has one white allele and one black allele. We call this incomplete dominance because you see the speckled chicken, it's mostly black, but some of that white is still getting through. So black is incompletely dominant over white for this species. We can also see codominance. So now this is where alleles are actually equally expressed. The easiest example to understand is in blood types. If you are type A, you have these type A receptors on the outside of your red blood cells. If you are type B, you have type B receptors on the outside of your red blood cells. If you are type AB, you have both. So here is one of the few examples of true codominance. And if you were type O, you just don't have either. Typo was actually originally written not that mathematical symbol meaning nothing, but somewhere along the way, someone misread it and it was transferred into typo, and that's what we still call it today. We see some systems of alternate dominance, especially when we're talking about flower color of, things that aren't peas. Because remember, Mendel's garden peas were either white or purple. But we do see a fair amount of incomplete dominance, especially when we're talking about red and pink flowers. And where our pink here is our heterozygote with one red allele and one white allele. So, what are Mendel's laws and what do they mean?